Much credit,
This meeting is being recorded.
much credit belongs, of course, to the faces that you see. Professor Esmond Snell, with whom I postdoced, he was also instrumental in shaping my career to a large extent and honing my critical thinking skills, my execution skills, things of that nature. So there are a couple of take-home messages that I want to communicate in this regard. Professor Vaidyanathan mentioned to me when I joined his laboratory, he said, when you start a project, you always start it with a high degree of enthusiasm and optimism. But at some point of time, that optimism actually becomes very miserable and you go through a treacherous period of disenchantment. So the key to success is to make sure that you keep that period of disenchantment to the shortest possible time so that you can enter the realistic phase instead of lingering in the pessimistic phase of your execution. That has served me well. So I recognize that it is a factor of everything we do. It's a feature for everything we do, whether it is relationships or whether it is projects or whether it is investments. It is not uncommon to encounter the transition from optimism to pessimism. But at some point of time, you have to bring in realistic assessment of the situation and make decisions as to whether you want to stick with it or not. Professor Snell had one pearl of wisdom as I met him the first time, which was, hard work coupled with critical thinking and a curious mind. Those are the key ingredients of success, and I focus on nurturing that. And those are still the talents that have nurtured me. And I'm not going to say that I'm an Ashtavadani or any kind of a Shatavadani or Sahasravadani, Avadani in that regard. But those aspects of curiosity, critical thinking, and hard work, where chance favors the prepared mind, is a fundamental take-home message. Over the years, I have had the opportunity to interact and work with many people. One of the other things I have learned in life is that you not only learn from your own work, learn from the work of others, you learn from the work of elders, you learn from the work of youngsters, and that every moment has the potential to be a teaching moment. Next chart, please. I did want to say that I have had the distinct pleasure and honor of working with multiple people over the years. Some have gone on to win Nobel Prizes, which is also a fantastic accomplishment and a recognition. Some have become National Academy members. But every one of the people that I have interacted with, whether they are entrepreneurs or professors in academia or my managers, they have all served me well. And even today, I maintain many of those relationships across continents. So while it takes a village to raise a child, it takes continents to sustain that. And certainly one should focus on making an impact. And one of the things that I learned from my boss, Professor Phil Needleman, who used to be at Monsanto, is that life is too short to be focused on projects that have a very small impact. So if you're going to spend your time, which is the most critical commodity that we all are blessed with, spend that time on working on interesting projects that make a huge impact on society. And my other boss, Professor Will Carpenter, who was also a professor and became the chairman of the International Chemical Chemical Weapons Treaty, after his retirement from Monsanto, he used to say, Kish, we are all in the business of making a lot of manure. He didn't use the term manure. I'm paraphrasing what he said. But make sure when you are in the business of manure, chase elephants, not mice. So those messages have stuck with me over the years. And it is something that I bring to the table every time I work with other people, because I don't do much work on my own these days other than the thinking part. Next chart, please. I must say that the revolution in DNA biology and all of molecular biology and biochemistry certainly began with the work of Watson and Crick, but it preceded Watson and Crick by almost 200 years by at least 150 years by Gregory Mendel, who made some fundamental observations regarding genes. This was all derived from plant biology, which shows the connection between plants and genetics, which laid the foundation, which has now been extended to humans, to bacteria and other living forms. But our ability to read, write, compose, and edit DNA is at the heart of much of the progress we have made and will continue to make over the next several decades in biochemistry and biotechnology. I must also mention that this progress in DNA has led to substantial progress in our ability to read, write, compose, edit RNA, read, write, compose proteins. And this is really the opportunity that we have as we transition from what I call single chemical reactions to living systems, where living cells become actually the factories, living cells become the delivery systems for biochemical activity, which is where our emphasis is today. And as we read DNA and write DNA, our ability to make DNA and make the DNA inexpensively, make that RNA inexpensively, is also at the heart of the biological revolution that we are experiencing. In fact, I must also say, since we talked about computers a little while ago, that the writing the genetic code is not that different from writing the software that people are writing. In Monsanto and in my own life, I realized that when we write the genetic code, that genetic code has to be in an appropriate hardware, and that hardware ultimately decides how that software expresses itself. And I'm now really pleased to see, with the evolution of augmented intelligence, which is the actual definition of AI, or the acronym for AI, AI has started moving in the direction of chip making in addition to writing the software, because hardware and software are contextually intertwined. Anyway, the next chart basically tells you why life sciences is such an important discipline of sciences, why this has such a huge impact on our lives, because feeding the world, healing the world, fueling the world, cleaning the world, protecting people in the world, protecting our ecosystem, protecting our natural resources, these are all dependent upon life sciences. And life sciences is, in my books, if I were to use the baseball analogy, it is in the first inning of a 9-inning game. Or if I were to use the cricket analogy, it is still the opening batsmen who are playing. There is an awful lot of knowledge that we are yet to unearth and leverage for making our economies leverage biology fully. Next chart, please. When I joined Monsanto, I realized that although I was trained to a large extent in microbiology, chemistry, enzymology, and so forth, I had the opportunity to work on plants. In fact, Ernie Jaworski offered me to become the head of plant biochemistry. I said, I haven't studied plant biochemistry. So he asked me to meet a professor at Washington University in St. Louis called Professor Warner, who's very famous for his work in plant biochemistry. He wrote down a very simple equation on his board as soon as I went to see him. He said plant biochemistry minus photosynthesis equals 0. And he asked me if I had taken courses in photosynthesis, and my response was yes. And he said, "Well, there is not much more to learn. You're good enough to lead the plant biochemistry program at Monsanto." And that's how I got into plant biotechnology. Of course, Professor Warner was trying to humor me, and over the years, I learned a lot more about plants. So it was learning on the job that really shaped my life, and I was able to move from microorganisms and chemistry to the discipline of plant biotechnology. I don't have to spend much time on this chart other than to tell you that our civilization began 10,000 more than 10,000 years ago with agriculture as the foundation for civilization. Even today, if we have to manage and navigate the continental divide that we have, the national divide that we have, the regional divide that we have, I think agriculture and plant biotechnology has a long way to go. And with plant biotechnology, we have the potential to not only address our food and feed needs, but also other needs, as I have shown on this particular chart. And I must also mention to you that many times we go to resorts and recreational facilities and so forth. What charms us are the trees and the flora, the fauna in that region. In addition to the mountains, the lakes, the rivers, no one wants to visit dirty lakes, dirty oceans. If you have algal contamination of beaches, no one wants to go be there. So in order to clean up many of these things, including cleaning up our atmosphere, biology, especially plant biology, is critical. Next chart, please. When I started working in Monsanto, my own career began with two different molecules. One which I talk about a lot, the other which I haven't talked about very much, but was foundational to the work that I did on glyphosate, which is the active ingredient of a herbicide Roundup, which was discovered at Monsanto, not as the molecule, but the molecule with the activity. There is a story about glyphosate, which I will tell you in a bit, but let me first spend a few minutes on NutraSweet, which was also another molecule which Monsanto acquired by buying GD Searle. Both of these were serendipitous discoveries. Chance favors a prepared mind is what I mentioned to you. It turns out that people who were interested in making NutraSweet were not interested in making NutraSweet as a sweetener, but they were interested in making an anti-ulcer drug candidate called gastrin. They wanted to determine, this is scientists at GD Searle, wanted to determine if gastrin had anti-ulcer activity because this was a hormone that was produced in the stomach. The first two amino acids of gastrin are aspartic acid phenylalanine. They made aspartic acid phenylalanine ester, methyl ester, and the chemist who made it accidentally put his fingers as he was putting the stuff into his NMR tube and found that his fingers were intensely sweet. And then he decided to taste a small crystal of NutraSweet. It was intensely sweet. He told his boss asked him to go away and focus on making gastrin. But then his boss's boss realized that there was a need in the planet for a non-nutritive sweetener, which was not called a saccharin, which was not called cyclamate, because at that time, saccharin had huge issues with respect to its potential for inducing cancer, and cyclamates did not have a good taste, and neither candidate had a good aftertaste in the sense that one had a metallic and the other had a bitter aftertaste. So I spent the first 2 years at Monsanto trying to figure out how to make NutraSweet. And our team was able to reverse every biochemical reaction we had studied in school. Aspartase, an enzyme which takes aspartic acid to fumaric acid and ammonia. We reversed that reaction by taking ammonium fumarate and making L-aspartic acid. Phenylalanine ammonia lyase, which converts cinnamic acid to converts phenylalanine to cinnamic acid and ammonia. We were able to take ammonium cinnamate and make L-phenylalanine from it. And we were able to use proteases to couple aspartic acid and phenylalanine and make aspartyl phenylalanine methyl ester. So people thought that we were wonder kids, and that was the reason why I was recruited to head plant biochemistry, because in a matter of 6 to 12 months, we had demonstrated that we could make this molecule, not only make the molecule, but make it at a price point around $60 a kilogram, whereas our competitors in at that time, GD Cell, which had discovered the molecule, was not capable of making it for less than $150 a pound, not a kilogram. So that gave us the freedom to operate, so to speak, and that led me to focus on the molecule glyphosate, which is the active ingredient of Roundup. The discovery of Roundup itself is a case in serendipity. Scientists in Sanders had made the molecule in 1952, tested it for various applications, but their assays never ran too long, in the sense that their herbicide testing was only for 2 days, and there was no herbicidal activity that they could see with this molecule in 2 days. All assays in Monsanto ran for 7 days for herbicidal screens. And when glyphosate was tested, it was tested on a Thanksgiving week. So the assays actually ran for 9 days instead of even the 7 days. And lo and behold, they found that every plant that was treated with glyphosate was killed. Although there was a lot of interest in making selective herbicides, no one wanted to kill corn, no one wanted to kill soy, no one wanted to kill the crop plant with a herbicide. But this herbicide did not distinguish a crop plant from a weed. It was a non-selective herbicide. So when chemists told their boss that they had made a non-selective herbicide, they wanted the chemists to add methyl, ethyl, and what I call futyl groups, so that those derivatives of glyphosate could be made more selective. But none of those molecules actually worked, and we ended up with a non-selective herbicide, which was used in clearing vegetation, which was registered in Malaysia, Brazil, and a number of other countries. Its mode of action was not understood at the time it was made. Monsanto ended up with patents on the use of glyphosate and salts of glyphosate, because Monsanto had a very clever intellectual property lawyers who were able to figure out how to claim compositions of matter, because the acid was what was described by Sanders, and it was the salts of glyphosate that were herbicidally active. So that is the long and short of the serendipity that surrounded the discovery of two important molecules. Roundup has probably made more than $150 billion now, as a herbicide, and NutraSweet probably has generated about $15 to $20 billion as a non-nutritive sweetener to various players. Next chart, please. As I mentioned to you, Roundup is a non-selective herbicide, kills all kinds of plants. So the project that I was asked to focus on was to make crop plants, especially crop plants like soybeans, corn, withstand the herbicide. We had advanced our capabilities in plant biotechnology, where we could introduce and express genes in plants at that time. We were searching for applications. Obviously, we had Roundup, which was a non-selective herbicide. The task was to make this non-selective herbicide spare the crop plant and kill the weeds. It had all kinds of advantages and that were going for it. So that was the task I was assigned. Next chart, please. I must mention to you that weed control is actually central to agriculture. Many of you have farming backgrounds. I do not know how many of you actually have worked in a field. And my grandfather on the paternal side used to farm, and I used to go when I was about 8 years old, 9 years old, to farm during summer holidays. And he used to take me to the field and had me remove the weeds. It was mind-numbing, back-breaking work. And in fact, weed control, if you were to add up the time that humanity has spent on weed control, since the dawn of agriculture. That number exceeds the number of hours humans have put in any other activity. Weeds actually compete with crops, lower the quality of crops. They compete for nutrients. They compete for water. They compete for land. They compete not only in those regards, but they also lower the quality. Many of these weed seeds lower the quality because they contain toxicants. Um, uh, and uh, and of course, they harbor pests which can attack croplands and so on and so forth. The long and short of it is we control weeds in agricultural fields not to make the fields look pretty, but because weeds actually are detrimental to both productivity and quality. Next chart, please. So the task of engineering glyphosate tolerance into croplands was the task that I had to spearhead. And as any self-respecting biochemist would do, I said there are 4 possibilities, theoretically at least. One is to overproduce the target enzyme. We didn't know what the target enzyme was at that point of time. Second is to mutate the target enzyme so that we could reduce the sensitivity to glyphosate. Since we didn't know the target enzyme, we didn't know what to focus on and to mutate them. And of course, we also knew that we could degrade glyphosate because glyphosate was degraded in the soil. So we could isolate these enzymes that were responsible for degrading glyphosate, isolate those. We could consider inhibiting the uptake of glyphosate because we knew glyphosate was a systemic herbicide. It was not only active at the site where it was placed on the plant, but it also moved up and down, meaning that it moved to the roots and shoots of the plant. So we could also consider inhibiting the translocation of the herbicide from the site of application to the sites where it could act. So these were some of the possibilities we drew up on the board. Next chart, please. The next chart shows the pathway that we knew that glyphosate acted upon. It is a pathway that has stuck with me. I do not know why, but sometimes in nature, sometimes in life, life actually has a reason why you study aromatic compounds. So in Dr. Vaidyanathan's lab, I studied aromatic amino acid metabolism. And in Snell's lab, I studied pyridoxal phosphate metabolism and pyridoxine metabolism. And in Monsanto, I made aspartic acid and phenylalanine and combined them. Phenylalanine is, again, an aromatic amino acid. And of course, Roundup act on the aromatic amino acid pathway that was known in the literature. And the work that we did and some of our academic collaborators did demonstrated that glyphosate worked by inhibiting an aromatic amino acid pathway enzyme called enol pyruvyl shikimate 3-phosphate synthase, which is shown here. But since I don't have a pointer, I can't point it to you other than to say that it was converting shikimate 3-phosphate to enol pyruvyl shikimate phosphate. Turns out that we knew a little bit about the bacterial pathway. In fact, P.R. Srinivasan, a professor at Columbia, working with Princeton and Columbia University, had deciphered the aromatic pathway for the biosynthesis of aromatic compounds. Yeast also biosynthesizes aromatic compounds. We humans cannot make aromatic compounds. We derive most of our aromatic compounds in our diet. This is a pathway which accounts for about 40 to 60% of the carbon that is fixed in plants. And of course, in plants, very little was known about the aromatic amino acid pathway. Next chart, please. The specific reaction that is inhibited by glyphosate is the reaction that is shown on the top here, called enol pyruvyl shikimate phosphate synthase. The 2 substrates, shikimate phosphate and phosphoenol pyruvate, as you can see, the arrows are being moved by C.S. Ramesh. Thank you. And you form a tetrahedral intermediate that is shown, and that intermediate disintegrates to make enol pyruvyl shikimate phosphate. There is one other analogous reaction in nature These are the only two reactions that I know of where phosphoenol pyruvate is actually used as a carboxyvinyl moiety donor. And the second reaction is in the biosynthesis of UDP N-acetylmuramic acid, which is an important component of bacterial cell wall. And that is the reaction at the bottom. But the PEP analog, which is what glyphosate is, it does not inhibit this reaction. And the next chart shows the two molecules which are very specific. These are both PEP analogs. One is glyphosate, which has strong herbicidal activity, and the other, which is shown, is the molecule of which is the phosphoglycidic acid, and this particular molecule is capable of inhibiting gram-negative bacteria. In fact, you can make spheroplasts if you treat the gram-negative rods with this particular molecule. So, in fact, glyphosate could not be used as an antibiotic for that reason. And the glyphosate became a fantastic herbicide, generating billions of dollars, and has made life very simple for a number of people engaged in farming because it kills weeds. It can be used in what is called no-till agriculture, etc. But the task of engineering glyphosate tolerance was no easy process for us, especially in the early 80s when I started working on the project. Next chart, please. I will summarize a lot of years of work in these charts. We found that we were able to isolate glyphosate tolerant plant cells. We were never able to make mutants of plants, whole plants, but we were able to make plant cells, which were able, Rob Fraley at Monsanto did this work. He isolated these mutant cell lines, and those mutant cell lines I demonstrated had higher EPSP synthase activity. So we knew that overproduction of the enzyme, the target enzyme, and that was capable of conferring glyphosate tolerance. And we use this overproducing cell line as the source material for isolating the protein, for isolating the cDNA for EPSP synthase. The cDNA for EPSP synthase was isolated by Dilip Shah, one of my coworkers, and we were able to demonstrate that EPSP synthase from both bacteria and plants were very similar. E. coli was the bacterial EPSP synthase we had at that time, and the plant EPSP synthase was similar, except that it had 72 extra amino acids at its N-terminus. And we demonstrated that these 72 amino acids at the N-terminal region was responsible for translocation of the EPSP synthase from the cytosol where the protein is made to the chloroplast, which is where aromatic amino acid biosynthesis occurred. We also demonstrated that pre-EPSP synthase was enzymatically active. Pre-EPSP synthase could convert shikimate phosphate and PEP in the cytosol to its product, although we knew that there was not much shikimate phosphate in the cytosol. And we demonstrated that the 72 amino acids sequence was capable of transporting other proteins, serving as a signal peptide. It was not dependent upon EPSP synthase coding sequence. It served as an independent moiety on its own. We did some of the most pioneering work on chloroplast protein import at that time. And we, of course, did the work of engineering the EPSP synthase into plants, overproduced them, demonstrated they were glyphosate tolerant. But as my boss told me at that time, tolerance is a very generous term to describe these plants because those plants were commercially irrelevant. Next chart, please. We also solved the crystal structure of EPSP synthase in 91-92 timeframe. It has a very interesting Pac-Man-like structure, 427 amino acids. The N-terminus packs into its own Pac-Man-like structure. The C-terminus has its own structure, but the crossover occurs at a couple of different regions in the protein. The first 90 amino acids or so in the N-terminus, and then around 200 to 250 amino acids are back in the top domain, and then the rest of it is all in the bottom domain. And the beauty of this Pac-Man structure is when shikimate phosphate, the substrate binds, the Pac-Man bites that, and glyphosate can bind it, or phosphoenol pyruvate can bind it. Next chart, please. And that, of course, results in a closed structure. So the task I had was to determine how much overproduction was actually needed based on our understanding of enzyme kinetics, based on our understanding of Km, Vmax, or Kcat, of the substrates, the products, the Ki for glyphosate, and so forth. And I drew up on a chart the overproduction that was required in order to use the wild type enzyme, and concluded that we needed to overproduce 10,000-fold the EPSP synthase protein to make glyphosate tolerant plants. 10,000-fold overproduction of EPSP synthase means that we should produce more of this protein than even Rubisco, which is a tall order. So I don't have to tell you more about this. We decided that we should focus on making glyphosate tolerant EPSP synthesis. So we selected bacteria, and one of the beauties of working with both Vaidyanathan and Snell's lab is we had a combination of chemistry and microbiology. And I liked microbes because I could get results within a day or two, sometimes within a week. Whereas with plants, it would take forever to get results. By working in bacteria, we were able to make E. coli cells that were capable of withstanding glyphosate. Most of those overproduced EPSP synthase because glyphosate was not very effectively imported. But I was also able to isolate some mutants that resisted because they had resistant enzymes. And one of those resistant enzymes we determined had a mutation in the glycine 96 position, which was mutated to an alanine. And that particular mutant, called the GA mutant, is the one that we initially advanced for purposes of engineering glyphosate tolerance. Within days of isolating that mutant, I determined the Km for PEP, the Ki for glyphosate, and concluded that although it was a great enzyme with respect to glyphosate tolerance, it was a lousy enzyme when it came to catalyzing the EPSP synthase reaction under physiological conditions, because my assumption was that the Km for PEP actually was indicative of the physiological concentration for this particular molecule in the chloroplast where the reaction occurred. So we defined the characteristics of an ideal enzyme, went in hunt for that enzyme, both by further mutagenesis of the glycine to alanine mutation that we found, as well as by going into the manufacturing plant of glyphosate and isolating from the waste stream where we had the discharges microorganisms that could grow in the presence of glyphosate. One of those microbes turned out to be an Agrobacterium CP4 species, and that had an EPSP synthase that had the near ideal characteristics for glyphosate tolerance. Interestingly, this CP4 enzyme also had a glycine to alanine mutation, or I should say it was naturally probably present. And when we replaced that alanine with glycine, that became more glyphosate sensitive. So it was very interesting to say that the mutation we discovered in E. coli was also true in the case of Agrobacterium in terms of glyphosate sensitivity. But these enzymes had very little homology with the E. coli enzyme or the plant enzyme, The plant enzyme and the E. coli enzymes we refer to as class 1 enzymes, and the agrobacterium and pseudomonad enzymes we refer to as class 2 enzymes. Next chart, please. It turns out that the work that we did at that time is still true today, and most of the enzymes fall into two classes, class 1 and class 2 enzymes. Class 1 enzymes within them have about 70% homology at protein level, 60% plus identity. Class 2 enzymes have less than 50% identity at the amino acid level. And homology is typically below 55%. These are generally more glyphosate tolerant than the class 1 enzymes, but the Km for phosphoenol pyruvate for all class 2 enzymes is generally very low, which means that these particular enzymes have evolved to withstand the herbicide. Next chart, please. Shown here is the X-ray crystal picture of the Pac-Man structure that I showed you before, which shikimate phosphate that is bound. It is interesting that the Pac-Man closes into the closed structure once the shikimate phosphate binds. The glycine residue 96 is actually at the gateway, at the very mouth of the Pac-Man. As it enters, if you move the arrow to the extreme right, that is where the glycine, the alanine serves as a hindrance for the glycine to come into the active site. And of course, we have now the X-ray pictures to validate. And of course, we can do additional engineering as well. Next chart, please. I mentioned to you that the aromatic amino acid biosynthetic gene, the EPSP synthase, has 72 extra amino acids at the N-terminus. Since that time, all the pathway enzymes have been cloned, and we now know that the entire amino acid pathway occurs in the plastids. And in order to take the bacterial enzymes and engineer high levels of glyphosate tolerance, we had to use this chloroplast transit peptide along with the bacterial EPSP synthase, so that within the cell, it would find the right locale for catalyzing the EPSP synthase reaction. Next chart, please. In addition to working on aromatic amino acid biosynthetic pathway, one of my other hobbies was to isolate microorganisms that would degrade glyphosate. Working in conjunction with my esteemed colleagues at Monsanto, we were able to isolate a number of organisms from the waste stream of the glyphosate manufacturing plant by screening the ATCC culture, etc. Within a matter of a couple of weeks, we were able to demonstrate that there were two pathways using solid-state NMR, where we were able to label the glyphosate with C13 and N15, either on the phosphonomethyl side of the glyphosate molecule or the glycine side of the glyphosate molecule. And we were able to grow the organisms on labeled C13 and 15 labeled material using solid state NMR. We demonstrated that this could either go into sarcosine, which is the bottom half of the pathway. From sarcosine, it could go to glycine, or as is shown on the top, it could go via the cleavage of the glyoxyl moiety to make glyoxylate and make aminomethyl phosphonic acid, which could then be cleaved to methylamine using the CP lyase. CP lyase is an enzyme that cleaves the carbon-phosphorus bond. This is an extraordinary biochemical reaction. It is carbon-phosphorus bond is very similar in terms of bond strength to carbon-carbon bond. So to cleave, this was no easy task. And I started characterizing CP lyase enzyme in crude extracts. And indeed, I was able to detect the activity, but I was never able to purify the enzyme. And of course, we had to use genetics, and genetics demonstrated that there were at least 7 pathway genes. And subsequently, next chart, please. CP lyase reaction was, The CP-Lyase reaction was demonstrated to be an S-adenosyl methionine-dependent, ATP-dependent reaction, where the ATP-dependent reaction initially adds a pyrophosphoryl moiety to a ribosylated derivative of the phosphonate. And this molecule then uses the S-adenosyl moiety to make a methyl radical, as well as, of course, phosphate. So a very complex reaction. 7 to 13 genes are involved in the pathway. We decided that this was great for studying the mechanism of the reaction, but it was not for engineering into plants. It was a decision that I had to make at that time in Monsanto. And of course, it was a good decision because I think it would have taken us 25 years if we wanted to do this via plant biotechnology. Next chart, please. So the two pathways that have served us well and have served us well through the eons since the 90s, mid-90s, when we made glyphosate-tolerant plants, are the bacterial enzymes, which we were able to target to the chloroplast. So these were glyphosate-tolerant TPSP synthesis. And the other pathway, which is the pathway that I did not go to in detail, the enzyme glyphosate oxidoreductase, which is capable of converting glyphosate to aminomethylphosphonic acid and glyoxylate. This is a flavoprotein. The enzyme is called GOX. It turns out that aminomethylphosphonic acid itself is a weak herbicide, so we have to detoxify AMPA by adding an acetyl group, and that makes N-acetyl glyphosate. So quite a few plants are sensitive to AMPA, and we had to use a combination of GOX and glyphosate N-acetyl transferase to engineer tolerance via this mechanism. Next chart, please. At the same time, we were doing this work, which was mind-blowing, back-breaking work. We also had a team in Monsanto led by Rob Harsh, which was focused on understanding how agrobacterium in nature is capable of making plants tumorous. So agrobacterium is one of those bacteria. When it infects plants, it gives tumors to plants. And of course, nobody wants to give tumors to crop plants. But by understanding the biology underlying this process, the work that was done by Mary Dale Shilton at Washington University, Gene Nestor at Washington University in Seattle, or University of Washington in Seattle, Jeff Shell, Van Montague, a number of other people, they demonstrated that there was a TI plasmid, and then there was the agrobacterial chromosome. The TI plasmid, which is shown in the bacterial rod cell at the top, left-hand part, was introduced into plants. A particular region called the tDNA was introduced into plants when bacteria came in contact with cut surfaces of plants. This tDNA carried a number of phytohormone production genes. There were auxin and cytokinin biosynthetic genes, and that is what produced the callus tissue, which you see on the plant and on the Petri plate in the bottom two parts. The goal in Monsanto was to disarm this tDNA, taking this TI plasmid, removing all the phytohormone genes, replacing that with genes of interest. Next chart, please. Two systems are being used today to make transgenic plants, and we took advantage of that. One is the particle gun bombardment technology, where you literally shoot DNA on tungsten projectiles into callus tissue or regenerable plant tissue. This is a technology that is now being used to make a lot of plants. The disadvantage with this technology is that the efficiency is very low in relationship to what is shown on the left-hand side using the tDNA. And there are two different technologies. One is a binary technology, where we use binary vectors, where the tDNA itself was on one plasmid, and then the other plasmid carried the genes of interest. One was a gene of interest in terms of herbicide selectable marker or a herbicide trait of interest or an insect control gene, and the other was a bacterial selectable marker, which included selection of either the plant cells or also the bacterial cells to ensure the presence of the plasmid. The most expensive tool of the plant molecular biologist is shown at the bottom, the paper clip, which is what is used to make explants of plants. We start with a leaf tissue, punch out the leaf disc, and then expose the cut surfaces of the punch leaf to agrobacterium, which has the competent genes, which has the competent plasmids, genes of interest. And then, of course, we apply the selection pressure. If the selection pressure is glyphosate, that is wonderful, but glyphosate is a very poor selectable marker. We had to use antibiotic resistance for selection, and then we scored the plants or selected the plants using glyphosate tolerance as the mechanism. I must mention to you that the genes that we introduce into plants or into eukaryotic systems obviously have a promoter region upstream of the coding region, and we also have a polyadenylation signal. So the genes that we introduced from the CP4 EPSP synthase that I described earlier had plant promoters that we isolated from viruses. The cauliflower mosaic virus 35S promoter, which is a viral promoter, the figwort mosaic virus promoter. We had the 72 amino acid targeting sequence that I mentioned, a cleavage site to cleave the signal peptide, then the CP4 EPSP synthase, a polyadenylation signal to cause the polyadenylation of that mRNA that was made. Those are all the genes that was the entirety of the gene that we introduced into the agrobacterium vector and subsequently to plants. Next chart, please. So we made the transgenic soybean plants using particle gun bombardment technology. At that point of time in the late 1980s, agrobacterium was still not developed for soybean transformation. One of our partners, AgroCetus, had developed the technology, and we worked with them to make about 132 transgenic plants for a price tag of $1.5 million. And so you can imagine every transgenic plants had therefore an expense associated with it. And if it failed, it had to be literally pitched in a dustbin. So I was not ready to throw dollars into the dustbin that easily. And I hedged as much as possible, so much so my boss at that time, Rob Fraley, said I was being too faint-hearted for being the leader and making choices. But nevertheless, that is what ultimately paid off in the end, because I hedged and hedged for every transgenic plant that we made. We made a large number of plants which had EPSP synthase genes in combination with the GOX gene that I mentioned earlier, without the acetyltransferase. We made a large number of plants which also had the EPSP synthase gene alone. And we only had about 32 plants because the 132 plants had to be divided into various vectors that we use for transformation, because we did not have any more money to spend on transformation after that point. 2 plants survived Roundup treatment at 32 ounces per acre. That is 2x the dose that actually killed the plants, soybean plants. Soybean is actually more difficult to control than most of the wheats. So there was a natural degree of tolerance. And every plant that had the GOX gene, the glyphosate oxidoreductase gene, was even more sensitive to glyphosate than the ones that had the EPSP synthase gene. Only 2 plants survived. One of them we advanced for commercialization. So the outcome of $1.5 million expense was 1 plant that we could advance for commercialization. Next chart, please. In order to commercialize the plants, we had to go through a bunch of regulatory clearances, field tests to demonstrate that they performed as advertised. And indeed, all of those hurdles were met. But we made one big mistake at that time. We ignored what was happening in UK in the context of mad cow disease. UK was very advanced in plant biotechnology at that time. And the UK science minister made a promise on television saying that mad cow disease had nothing to do with proteins. It had nothing to do with human diseases, especially wasting diseases in the brain, the scrapie disease, as it was called. That although it caused the mad cow disease in cows, that disease could not be inherited by eating mad cow disease infected meat. It turns out that these proteins, which are called the prions, are capable of infecting, entering mammalian cells, infecting humans. And that when came out, of course, trust in plant biotechnology declined. Prince Charles started blaming Monsanto, started blaming all the plant biotechnology workers and said plant biotechnology has no place. We should be resorting to age-old technologies for farming. And of course, that meant I had to fight more acceptance battles on plant biotechnology. We came up with a regulatory protocol where every country on the planet had to approve plant biotechnology products, although the prescription was worse than the disease. Nevertheless, that is what we ended up with. And we had to go through the regulatory process, which took us a couple of years in the United States. And then we had to clear from importation of those seeds in China, in Japan, Russia, number of other countries. And even clearances sometimes were delayed. And we had to work through the logistics of soybean cultivation to make sure those countries did not receive those products. The long and short of it was I started off as a biochemist, ended up as a general manager, including meeting people in India. And I remember meeting Mr. Mishra, who became an agriculture minister because the opposition had taken over, the BJP had taken over at that point of time. It was called a loose coalition of sorts with the Communist Party and other leaders. And Mr. Mishra said to me, Dr. Kishore, you represent a capitalistic economic principle coming from Monsanto and coming from a country that adheres to that principle. I'm a communist. You might wonder what is in common. Let me assure you, communists are actually capitalists, except that we want that capitalism to reside in the country, not outside the country's kings, queens, bureaucrats. Very true statement. And in fact, that is true in China. And of course, Mr. Mishra was very supportive, as was the Congress Party. Rajiv Gandhi and Mohan Kumar Mangalam were very, very supportive of the technology. Monsanto actually offered the technology of BT cotton that I will talk to you about in a second. And to India at that time. But lo and behold, the politics reared its ugly head between Vandana Shiva and a number of other activists. Plant biotechnology never saw the light of the day as it should have in India at that time. Next chart, please. One of the greatest contributions of Roundup Ready technology to agriculture is not only better weed control, but a concept called no-till farming. I mentioned to you that agriculture came about 10,000 years ago, since the dawn of civilization. But at that point of time, what we did was to go to a particular region, use that for a particular period of time. If the rainfall was good, if the land was good, and once weeds started infiltrating those regions, either we would burn it if we had the resources to burn it, or we would simply go to the next area and then start damaging it. And we invented the plow. And of course, the plow system was fantastic. But while it provided for tillage of the soil, the aeration of the soil, which was good, tillage also, of course, weakened the soil structure, and we lost a lot of topsoil. And over the years, we have lost significant amounts of topsoil, not only because of erosion by air, but by wind, but also because of water. So no-till agriculture allows us to directly drill seeds into the land without having to damage the topsoil. If we want to aerate the soil, we can actually drill holes and aerate the soil, a very efficient technology. And glyphosate tolerance has actually enabled farmers to use no-till farming in a big way. And since its introduction, indeed, in the United States, farming has moved from tillage farming to no-till farming, which enhances, of course, the quality of the soil. Next chart, please. I don't want to spend any time on conservation tillage. There are a number of other benefits, especially at this time of greenhouse gas concern. It conserves and holds the greenhouse gases in place. Tillage actually destroys the organic matter that is present in the topsoil due to the metabolic activity of the microbes, in addition to, of course, the erosion by water and the wind. Next chart, please. Shown here is the impact of Roundup Ready technology. In Brazil, they used to struggle to grow 2 crops of soybean in a cropping year because these are moderately tropical regions in Brazil. They are able to grow 2 crops in many regions, and in the tropical regions, they can grow 3 crops. What it enabled the farmers in Brazil to do, that is the introduction of Roundup Ready, is to harvest the soy and come back with a planting of corn, because both crops were made Roundup Ready by using the genetic technologies that I described. And today, Brazil is able to get 3 crops in one annual year, and in Uruguay and a number of other places. So farmers can use their land depending upon the quality of the soil and the other considerations. And this has enabled Brazil to become a major exporting country for soy and even corn. Next chart, please. I don't want to take much time other than to mention to you that the work that we did in the 90s is now being repeated in the academic circles. A lot of that work is now been followed on in academia, in bacteria. People have confirmed all the work. Of course, we would never have been able to make glyphosate tolerant crops if our work wasn't good. But nevertheless, the theoretical aspects of what we concluded has also been demonstrated. Today, people are engineering resistance in bacteria, especially the class 1 EPSP synthase containing bacteria. People have studied also glyphosate degradation, phosphonate degradation, et cetera, et cetera. Next chart, please. The impact of commercialization of glyphosate tolerance means that farmers started using glyphosate as the only herbicide. Although we advise farmers to mix and match herbicides, of course, farmers do what they want to do. And that impact has continued to result in emergence of glyphosate-tolerant weeds. In addition to glyphosate-tolerant weeds, other herbicides also have this issue. Shown here are the incidence of weeds, and also the discovery of new herbicides basically took a backseat, so to speak, because no one was interested in competing with glyphosate at the price points that it was being offered. So it basically made it an unattractive proposition until a few years ago. Next chart, please. Shown here is the red line, which is the incidence of glyphosate tolerance or Roundup tolerance. Other herbicides also suffer from this problem. We are trying to understand, when I say we, not only scientists in some of our companies, but also Monsanto is trying to understand how weeds actually emerge. Most of this appears to be a selection pressure, Some of it is also, of course, new mutations in the wheat seeds that enable the emergence of these wheat seeds. Next chart, please. Boll guard cotton is another technology that was developed when I was at Monsanto by my colleagues, David Fishoff, Fred Perlak in particular. Cotton is one of those crops which uses a lot of insecticides. Nearly 40% of all insecticides that are sprayed today, that were sprayed at least until BT cotton came into the marketplace, were used in cotton fields in the United States and other places in the world. Cotton is actually very, very tasty to a caterpillar family of insects. And even today in India, there are pests that attack cotton plants. So this is a fiber that is, that is actually liked by many people. But if you don't have insect control, you can see the yield of cotton in the cotton that is harvested. In one hand, on the left side, you can see the amount of cotton that is harvested, but if you have insect protection, the amount that you can get. So lint yield goes up dramatically, the fiber yield goes up dramatically if you have the resistance. So what we did in Monsanto was to take an insecticidal gene produced by a class of organisms called Bacillus thuringiensis, and take the BT gene from this insect and engineer that into crop plants, especially cotton plants. Although that sounds very easy, in reality, it took about 5 to 10 years of hard work. We discovered that the BT genes that were present in bacilli could not be expressed in plants. So we had to design synthetic genes that were codon optimized for expression in plants. And not only they were codon optimized, but there were also regions which made the messenger RNA very unstable, and we had to engineer them out. This has all been published. So the synthetic gene was then engineered using the plant transformation vectors along the lines of what I described to make cotton plants that were resistant to the bollworm, cotton bollworm. Next chart, please. Since that time, the work has been extended to include maize, sugarcane, number of other crops. BT genes have been discovered that work not only on the caterpillar family of insects, but also the coleopteran or the beetle family of insects. Corn rootworm has control, which is essential in order to protect the root of plants. Stem borer, which is responsible for stem damage, earworm, which is responsible for damage to corn ear. All of these have, resistance has been conferred to crop plants by using BT genes that can kill these insects. And now farmers are growing crop plants that are using those BT genes, multiple BT genes, to protect the crop plant. In addition to the BT genes, RNAi also works against the boll weevil and the beetle family of insects. That technology is being advanced by Monsanto in plants, and one of our companies, the Greenlight Biosciences, is developing sprayed formulations and microbial formulations that can advance the technology. Next chart, please. We tried to bring BT technology to India in the late 80s, offered it to Indian government for $1 million, saying lock, stock, and barrel, you can do what you want to with this technology. There were people who asked for the technology for $50,000, and I had to walk away from those meetings saying, it doesn't even pay for the airfare of mine and the lawyers who have to work on the agreements. We were quite interested in bringing the technology to India, recognizing the plight of the farmers, because India has close to about 300 million farmers, as opposed to 1 million farmers in the United States. So we knew that this was important to lift the rural economy. To my delight, both Mr. Rajiv Gandhi and the prime ministers that followed him, Narasimha Rao, Vajpayee, every one of them was highly supportive of the technology, but the bureaucrats especially at the Indian Agricultural Research Institute, somehow had negative things to say, and it never really met the light of the day. And we decided to work with the private company Mahiko to bring the technology to India. And it is interesting, Mahiko gained approval only after a rogue cotton seed dealer called Navabharat Seeds decided to commercialize the technology by claiming that he had accidentally discovered insect-resistant cotton, and he commercialized that in Gujarat. Of course, it was no surprise that there was a BT gene, which was engineered by Monsanto in those plants. So BT cotton has been a remarkable success in India. I don't want to dwell on that other than to say India went from a cotton importing country to a cotton exporting country because of the introduction of this technology. Cotton is a heavily water consuming plant. And at one time I had to argue with the Congress science minister and the planning commission head that irrigation, while it is important for agriculture, is insufficient to make a plant produce the harvestable products, because insects and weeds and diseases compete for the same things that we are interested in. But be that as may, I spent more time pontificating about the technology than about working in the laboratory at that time. Next chart, please. We had to introduce the technology with a lot of care, creating refugia to augment the beneficial impact of the technology on the growers. So we set aside 10% of farmlands of the farmed area. In a country like India, that was very hard to execute because people only had an acre or half an acre, whereas in the United States, where they were cultivating 500 acres or 1,000 acres, it was easy to ask them to set aside 10 acres or 20 acres of land. So it was very difficult to bring that. There we had to resort to the refugia that was provided by other crops that they were growing in the region. The whole idea was to make sure that there was population of wild type insects to mate with resistant insects so that there was dilution of resistant insects if it evolved. We expected this resistance to evolve very quickly. It didn't happen because of the refugia management technologies, and we were able to introduce multiple BT genes in order to manage this resistance issue. Even today, the refugia practices we set up at that time is in vogue in many nations of the planet. Next chart, please. I mentioned to you that genetically modified crops are now being grown only in certain regions of the world. Not all of the world is still taking advantage of the benefits of the technology. In fact, not in my backyard is the expression that I heard when I was in Italy, because they want to be GMO free. I asked them the question, what do they think is a GMO? Because the food that we eat, the microbes that actually infect us, the microbes that are present in the plants, the microbes that are present in multiple organisms, including the meat we consume, are all genetically modified. They keep evolving. Just because God or nature evolves it in a particular way, it doesn't mean that what we humans do is inferior to what nature does. Certainly, of course, we also had to be mindful that we could introduce either knowingly or inadvertently, products that are not necessarily good for environment or good for humans or good for beneficial insects. And of course, safeguards have to be in place to manage those. But nevertheless, significant parts of Africa, which still suffer from hunger and malnutrition, significant crops in India, which suffers from significant malnutrition and hunger, Pakistan, etc., still do not take advantage of this technology. Next chart, please. I must also mention to you that crops today do not have just one or two traits. We started with the introduction of glyphosate tolerance in soybean in 1995, Bt cotton in 1995 in the United States. Over the years, this has grown to a point where corn plants today have 12 to 15 transgenes in them. And that's also true for soybeans, where they have genes for insect control, herbicide control, sometimes multiple herbicide control, etc., herbicide resistance, etc. In addition to that, quality traits are also being introduced where oil modification has gained significant interest. Next chart, please. Over the years, we have learned that the domesticated crops are a form of genetic engineering. Yes, nature did the genetic modification. We did the selection. The teosinte corn that was the precursor of the modern yellow dent corn, which you see in the inset there, is vastly different. I know no self-respecting farmer of today wants to grow teosinte. Teosinte grows in Mexico in many regions wildly, but that became the plant of today, and that required significant genetic modification. No human was involved in the genetic modification, but lots of humans were involved in the genome selection. The tomato that we consume today, I think the cherry tomatoes are closer to the tomatoes you see in the other inset. Those are the wild Peruvian tomatoes that grow in the Andes. And those tomatoes have since been domesticated to make the Roma tomatoes, the San Marzano tomatoes, the cherry tomatoes that are ovuloid in shape, round in shape, etc. It's yellow in color. All of this has occurred because we have been doing genetic modification for quite some time. Tomato actually is a toxic fruit. In fact, it could not be consumed as it was, and domestication required breeding out the toxicants of tomato fruit. So are a number of other crops that we have commercialized over the years. We have had to breed out toxicants, indigestible materials in them. And today, we have the crops that we have because of our ability to do the genetic modification, whether through the biology that I described or through the biology of selection, which breeders have been practicing for quite some time. Next chart, please. Even nature's genetic engineering can be mimicked in the laboratory. Shown here are some of the mustards. Cauliflower and cabbage are both are very related to sursu or canola that we know of. Arabidopsis plant can be mutated through genetic engineering to make cauliflower type of flowers. It can be made into a cabbage-like fruit. It can be made into a radish-like root structure. So it tells you that the genetic engineering has the ability to recreate from new germplasm materials that nature did and we selected, because that potential opens up vast doors to improve our food systems. Next chart, please. I just want to tell you that humans, the early humans, could go from one region of the world to another region of the world when their farms were unproductive. Nobody had titles to those land. If you were stronger, if you had guns, if you had arrows, you could displace those people, occupy their land, and farm it and use it for producing food. If one region did not produce, you moved to another region which was capable of producing food. We do not have that luxury anymore. We have to rely upon the land that we have. I have news for you. If we want to bring more arable land, we have to cut more trees. Trees are responsible for taking out the carbon dioxide, which is a greenhouse gas in itself. And if we deforest to create more arable land, that is a kiss of death as far as humanity is concerned. So we need to make more agriculture more efficient, agriculture more climate friendly, all the technologies that I mentioned to you so far enable farmers to do exactly what I have described here in terms of making agriculture more sustainable. Next chart, please. When I was at Monsanto, we also worked on increasing starch content in a number of crops. We understood the rate limiting enzymes in starch biosynthesis, especially in potatoes. So we had the ability to increase the dry matter content of fruits, whether it is tomato or tubers, like it is potato, number of other seeds. We could use this enzyme, which was feedback regulated, taking away the feedback regulation of ADP glucose pyrophosphorylase. We could actually increase starch accumulation in the amyloplasts. Next chart, please. We could not only increase the starch content, but we could change the starch structure, making more amylopectin, which makes more waxy maize, for example, or more waxy wheat. We could make more amylose, which makes less digestible material, because during cooking, amylose makes indigestible fiber. So we could make a number of modifications in a number of crops using the technology for starch biosynthesis and starch modification. Much of this technology sits on the shelf today because of lack of GM acceptance in a number of countries of the planet. Next chart, please. Similarly, we were able to modify oils. Much of the work was done at Calgene. Work was done with Chris Somerville in collaboration with my group in Monsanto. We were able to make crops produce more medium chain triglycerides. Medium chain triglycerides are a very interesting class of nutrients when it comes to human nutrition. They are literally digested like sugars. In fact, many patients who suffer from Lorenzo's disease actually consume medium chain triglyceride because they burn the fat very quickly, the more digestible than the longer chain fatty acids. In fact, burn patients who have very high need for calories, medium chain triglycerides are used in burn patients to meet their caloric needs, especially for skin regeneration. Similarly, saturated fats, especially palmitic acid, which is a C16 fat, increases LDL cholesterol, which is responsible for cardiovascular imperfections that we see in people. So we can change the saturated fat profile. We can make some amount of saturated fat to meet the physicochemical texture that we desire in the food, the mouthfeel that we desire in food, but make more of the monounsaturated, less of the 18-2 types of fatty acids, which directly lead to the production of inflammatory molecules, and make more of the anti-inflammatory fatty acids like the 18-3 or other omega-3 types of fats, which actually reduce the degree of inflammation. Next chart, please. In addition to that, in collaboration with Chris Somerville, we started making polyhydroxy alkanoates, a class of polyesters. There is need to replace polyethylene and polypropylene in our system. We certainly don't have to replace it everywhere, but when in our oceans, in our riverbeds, and in our own body, we see polyethylene and polypropylene, that is cause for concern. And indeed, microplastics accumulate everywhere. These are molecules that are not easily degraded, and we need to replace them with polyesters, which are much more biodegradable. In the 90s, I started working in Monsanto on making polyhydroxy alkanoates. Indeed, we were able to make, and the next chart, please, a class of molecules called polyhydroxybutyrate valerate. This polyhydroxybutyrate valerate was made in the seeds of not only Arabidopsis, next chart, please, but also in canola. And they had about 5% of the canola seed actually had polyhydroxybutyrate valerate. The idea was to harvest the polyhydroxybutyrate valerate and leave all the other components as food components or feed components. Of course, because of the activity of activists saying that um, no food material can be derived from industrial uses, we decided not to pursue those programs. Indeed, we were able to demonstrate, next chart please, that many of the plastics that were made could be readily degraded either by composting. They were also capable of being degraded in landfills, but much of the technology sits in the shelf. And today there are companies in the next generation of biotech companies, industrial biotech companies, which are trying to make these molecules using fermentation. But I can assure you that they will never be able to compete with petroleum-derived materials. Only plants have the capability to compete with petroleum-made materials, and we can make them renewably. Next chart, please. We also introduced a bunch of genes into oil seeds. These are for purposes of increasing the carotene content. Indeed, we made a high beta carotene containing canola oil. We were highly interested in taking this to India and Nepal. Some of our collaborators and partners in this work, Ingo Petrikas and others at Swiss Institute, engineered this very similar technology into rice. In fact, golden rice, if you have heard of it, Ingo Petrikas has been trying to commercialize that with Syngenta. And in Philippines, it has been approved, but it is still not commercially cultivated. Vitamin A deficiency afflicts close to about 125 million people in India and Nepal today. And around the globe, it afflicts about 880 million people. In fact, in Nepal, when I talked to researchers there, they told me that one of the earliest symptoms of pregnancy is night blindness in women. And I said, why is that? And they said, well, now they have figured out that women of childbearing age are all vitamin A deficient, and when they become pregnant, they develop night blindness. And it is transitory, but there is absolutely no reason why women have to suffer from this, and why the diagnosis for pregnancy has to be night blindness instead of other technologies that we have on hand. Nevertheless, this technology sits on the shelf today. We can not only make the carotenoids of the type like beta carotene, lycopene, lutein, and so forth, which are important components of our eye tissue, but we can also make molecules like astaxanthin. We can make many of the saffron pigments that are present in our food using this technology. And if it is already built into the food, it is a foundational way of delivering via the seed the technology that can be used by farmers who don't have to depend upon any kind of a transportation system from urban processing centers to get nutrition. Next chart, please. I think about a year ago, I talked about increasing protein content by gene editing. That technology is being advanced, but that same technology can also be used to use, in converting plants to factories for making a series of plant fibers, a number of proteins or nothing but polyamides. In fact, nylon was invented in DuPont after studying silk. In the quest for making silk, they ended up making nylon, but we can go back to making nylon instead of growing mulberry plants and then putting silkworm. We can actually consider either in producing in mulberry silk proteins or in any of the other crops as well. We can consider making silk, we can make wool, etc., so that we can improve the sustainability of many of these industrially and humanly useful materials. Next chart, please. In the final few minutes, next chart, please. I want to mention to you that agriculture in the United States has advanced quite a bit, not because of anything that we had to be scared about or ashamed about, but because of deployment of technology and a system that promotes the use of this technology. I don't have to tell you what the productivity in Europe is, even in a crop like corn. Europe, where they grow corn for a longer period of time, France, which you can see here, lacks United States. This chart was actually made in 2010. If I were to make this chart today, Europe is behind United States by a significant number. By not deploying the technology, biotechnology, one actually damages the ability of growers to deploy modern technologies, to reduce their labor inputs, to make their soil more useful for a longer period of time, to increase their income, to create new opportunities for their businesses. Every one of those which we felt were going to be true, they have now been demonstrated to be true. And the work that has occurred by the antis, I'm afraid I have nothing good to say about them. Yes, a certain degree of awareness is critical. We have to be careful in introducing products, but not to deploy the technology forever is a crime against humanity. So I do want to mention to you that if corn is to be used for other purposes other than for purposes of feeding cows or eating directly by humans, we have to deploy these technologies so that we can make the raw materials that can feed the bioeconomy businesses. Next chart, please. People argue about sustainability. I think to the extent that you can build into the genetic system of a plant, potato tuber is what is shown here, and with very little of work, you are able to harvest potatoes and use those potatoes either for industrial uses or food uses. That is vastly superior to the conventional. If you can lift the chart up a little bit. Oops, in the PDF, I have lost the essence of what I wanted to say, where a chemical factory makes chemicals and fertilizers and the engineering equipment and all the other things. And then we have tons of input, which emit a lot of carbon dioxide, which is greenhouse gases. And then we had to process these potatoes, sort the potatoes, all kinds of labor involved. That is not sustainability. We have the opportunity to directly use sunlight and water and nitrogen fertilization to make not only the food that we consume, the feed that we need, but also feed the ecosystem, to make the industrial materials and energy, to clean up our atmosphere in a much more powerful way. Next chart, please. A simple theme that I want to leave you with is that nature processes about 210 gigatons of carbon on an annual basis. Today, greenhouse gas mediated warming and global warming is a topic of serious concern. Nature emits and captures significantly larger amounts of carbon than we humans produce by burning fuels. In fact, the amounts our nature processes about 770 gigaton equivalents of carbon dioxide equivalents, all anthropogenic emissions. If we were to add up, that adds up to about 50 gigatons. So you can see we are not even at 10% of what nature does on an annual basis. We can actually use the capacity of our land and our ecosystems in order to recapture that carbon dioxide and transition from the old carbon economy to the new carbon economy. If we are to deploy biotechnology across the board, not only in agriculture, but in forestry, in horticulture, recreational land, and so forth. And that, ladies and gentlemen, is the fundamental message that I want to leave you with. There is no sustainability. There is no negative emissions era if we do not take advantage of what plant biotechnology offers. Next chart, please. People still debate as to whether we are in a global warming period. People still debate whether the carbon dioxide content of the atmosphere and whether methane content of our atmosphere, nitrous oxide content of our atmosphere is responsible for the warming. I'm not going to sit here and tell you that greenhouse gases account for all of the global warming, but there is no question they do contribute to global warming. We have nearly doubled the global greenhouse gas content over the last 30, 40 years. Temperatures are no longer what they used to be. Even in 1979, at a period where there was more population than in the 1950s, and we went through the first baby boomer period, compared to what it is today, where temperature, we have accepted that at 2 degrees Celsius warming, we are on trajectory for that within the next 25 years. It sounds like this is far away. I may not be around in 2050 to see what the temperature is like, but certainly my children, my own grandchildren, and I'm willing to bet your children and your grandchildren will be there. And that is the legacy we are leaving behind. There are technologies that we have today created by the convergence of various technologies, including biotechnology, that we can leverage, and we should be spending our time and resources to indeed take advantage of that. Next chart, please. When I was in Monsanto, we tried to create a life sciences company. I think the world is ready for life sciences countries, life sciences continents, and a life sciences world. The combination of agriculture, industrial biotechnology, food and nutrition biology reduces our dependence on pharmaceuticals. Pharmaceuticals have a role to play. Pharmaceuticals can help us with respect to disease management, but wellness management is not a paradigm that we should be using pharmaceuticals for. We should be actually taking advantage of what nature offers us in terms of clean water, clean environment, healthy food, nourishing food, healthy lifestyles. And I think that is possible if we were to switch to a greater dependence on plant biology and microbiology in combination with human and other biologies and the digital biology systems to sustain the world. Thank you. I don't think I need to go over this chart. I mean, because they convey multiple messages, but all leading to the same endpoint. I'm happy to respond to any questions you might have.
Thank you,
Thank you, Kishore. That was a wonderful talk about
job.
how industrial biotechnology can be used for human upliftment. Thank you very much.
Thank you.
Wonderful talk, Kishore.
Wonderful talk, yes.
Thank you.
So great job, Kishore. I had a question for you.
Sure.
I mean, is transgene silencing any of major concern, or what steps are being taken to avoid?
So, great question, Muthu Krishnan. So, transgene silencing we can demonstrate in the lab. But when we select in the field our materials, we not only evaluate the progeny, but we also do the breeding and evaluate in crossbreeds. So we take them through multiple generations before we actually introduce the product, and we see absolutely no evidence. It seems to me that a little bit that I know in that discipline, it is more dependent upon the of the chromosome where the transgene is introduced. There are indeed sites where there is a high propensity for methylation and other types of DNA modifications. And yes, epigenetic silencing occurs in those regions. In fact, the science of epigenetics itself has been of interest to me. I've been working with companies to leverage epigenetics to bring in epigenetic traits as well. But I must mention to you that with regard to transgene silencing, we have now products for 30 years. We have not observed a single case of transgene silencing. I must also make one other important point. Friends of mine in the digital technologies say we are absolutely safe. You guys have to prove that you are safe beyond anything before we can commercialize products. But an iPhone actually has killed more people today than any of the seeds we have commercialized thus far, or any of the traits I've commercialized. You might have seen on television where people have walked into edges of lakes and rivers and fallen because they were not paying attention to what they were doing. People who have been hit by automobiles. That's an example of an iPhone induced or a smartphone induced tragedy. But people have committed suicide based on what they read on the internet. I mean, every technology has its own pluses and minuses. Abused, everything can kill. Misused, everything can kill. But the technology by itself should not be the focus. It is how we deploy the technology and what the products of the technology are that needs to be the focus. I can wax and wane about it, but the reality is, I do not believe that we have won public confidence over the years that we have commercialized the products, and there are activists who continue to occupy the airwaves.
Kishore, I have one question. This is on a kind of a offshoot of what you're saying, because I think use of these plants is one aspect, but if you look at the western parts of US, where Arab countries have bought out large plots of land, and the local governments have no control over what they grow and how much of the natural resources they utilize, they are growing a soybean crop and using up all the groundwater, and they just take it out to feed their cattle and their people. So how do you, if you make a crop very, very efficient at proliferating, that's the risk you run into.
So let me make the following observation. The crop you're talking about is alfalfa.
Alfalfa, right, and not soy.
Not soybean.
Yeah.
Yeah. Alfalfa is used as actually a cattle feed because it is a highly proteinaceous, biomass. And in fact, alfalfa hay is used even in the United States, where they actually dry the alfalfa on the farm and transport it. The answer to your question is, first of all, we need to think about the ways in which we can green the desert. Because if we can't green the desert, whether it is a Saudi or a Qatari, or whether it's some other part of the Middle East, they will certainly have to go to other regions to get their food. The second part of it is countries which are giving them the land, which are selling them the land, they can enforce rules. It's not like they can't enforce rules. And the third thing is, I do not believe that the Saudis have, at least I can talk about Saudi Arabia because I participate on advisory councils there. I used to be on the board of KAUST as well for some time, which is King Abdullah University of Science and Technology. One thing I can tell you is they are interested in actually deploying modern technologies. In fact, desert agriculture, improved by both molecular breeding and biotechnology, is of interest. Saudi Arabia imports a lot of biotechnology products from the United States. So the Saudis have, or the Middle East for that matter, really is not against biotechnology. It is just that there are some religious zealots who also have used biotechnology to make their case that Allah was against biotechnology. and of course, I don't have to tell you whether it is Allah or Jesus or Moses or Krishna. You can make them all against biotechnology, but the reality is what we have uncovered in this process. So the only answer I can tell you is that Saudi Arabia itself is a water deprived region. If they want to make agriculture and make a go at agriculture, desert agriculture requires very, very stringent water management practices. And they are interested in building their own agricultural system because nutrition security is the most important objective of any politician. We can talk about this, about all biotech, GMO, and a number of other things. If there is not food to eat, Arab Spring, the Yemeni crisis, the disasters of Bengal, major rulers and governments have fallen because of lack of food. So, we can stay without food maybe for a few days, but without food and without water, there is no way we can sustain ourselves. And that is why this is the most important objective for most of these top-heavy governments. I use the term top-heavy because I want to tell you that control is actually led from the top.
Yeah,
Thank
one
you.
other
Kishore,
question I have,
I think
quick one.
Karnik has a question. Okay.
Yeah, CS, can I just?
Yeah.
Yeah,
Go ahead.
one of the slides you showed, I was curious because the list of countries you showed, starting with the US in the per acre production, there is no mention of Australia. I was curious why.
Australia does not have enough water.
Ah, okay.